Joining us today. Let's just dive right in here. So today I'll be discussing a project with colon adenocarcinoma, or COAD. This is the third most commonly diagnosed cancer and is attributable to about 9% of all cancer deaths. In addition to having both copy number and mutation data that's available for this data set, we also have extensive annotation information. And this includes things like the presence of polyps at diagnosis, which is a risk factor, age at diagnosis, anatomic site of the tumor, histological type, tumor stage, and also overall survival. For evaluating this data set, we're using Nexus Software. The level 1 raw asymmetric cell data, this is the raw array data, is loaded into Nexus where it's pre-processed. I'll go into this in a little bit more in just a moment. From there, the log ratios are calculated, and then the data is segmented to identify regions of change. At this point, we can also load in the level 2 MAF somatic mutations, as well as any sample annotation that we might have. Once this is all combined together, we can now identify regions of change within their genes of interest, and then ultimately any sort of functional relevance. All of this data is available at the TCGA data portal. The URL is listed right here. So as I mentioned, there are several pre-processing steps that are occurring within Nexus. This includes matched paired analysis so that we can learn about only the somatic calls, so the calls that are only in the tumor, not in the normal. We also have systematic correction, ploidy correction, and gender correction. All of these topics have been covered extensively both on our blog and in several other webinars, so I'm not going to be discussing those in detail today. If you'd like to learn more about them, please visit our blog, which is at blog.biodiscovery.com, or visit our webinar page, biodiscovery.com/support/webinar. And so with that, let's just jump right into our Nexus project. So here is the Nexus COAD project. This is going to look very similar to what you would typically see when you first upload your data and it's been processed. So here we have a sample name, and these are just the names that the samples have in the raw data file. We have our data type for the copy number data, as well as the status. You can see they're all processed. We have the sequence variation data type and that it's been processed, the quality score assigned by Nexus, and then we have currently 2 other factors that were both used for the pre-processing. So we have the diploid regions, which is used for ploidy correction, and gender for gender correction. Excuse me. But that's all the information that we have right now loaded into our Nexus project. Of course, like I said, there's extensive annotation for this data set, which I currently have in my Excel spreadsheet, as I'm sure most of you have for your sample sets. So if we open up my Excel spreadsheet, I have a column here that's labeled sample that has also the same name that was used in that raw sample file that I loaded in. I have patient ID, which is the actual ID for that patient, not necessarily the same information. And then I have all of my additional information here. So I've got total non-silent mutations, age of diagnosis, anatomic site, presence or absence of colon polyps, histological type, microsatellite instability status, tumor stage, and then I have some survival. And you can see that for some of these I have all of that information filled in, but for other ones, only some of these have the information. And that's fine. But I want to take this table and I want to load it into Nexus. So the only requirements for us to do that are, one, that we need to make sure this sample name, this header with sample, has the same names that match my project back here. So these need to be the same. And then the other thing I need to do is make sure that I've saved this as a tab delimited text file, which I can do in Excel. So once I've done that, I can go here to Factors, Load Factors, and now I can select this tab delimited text file and hit Open. And what's going to happen is Nexus is going to read that file and match up the sample names and then add all of my new factors to the far right. So now you can see that my entire table has now been added in here. I can rearrange the order of things, so I can just click and drag if I want to see things in different places. That's fine. And what you'll notice is I have all that information in here. I also have my survival information. Now, in order to do survival, we need to have 2 different columns. the first column we need to have is called event, and it needs to be called event in your spreadsheet, or if you load it up manually, you need to have a column named event. And within this, we need to specify the samples that are either alive or dead, or we can say living or deceased. It's really important that we check to make sure we have our spelling correct on that. The second column that we need to have is a numerical column that gives us the overall length of time. And so here I've called it overall survival. You can give it whatever name you prefer. But I have overall survival, and I have it in terms of days. And so this is, depending upon when they joined the study, the amount of days they have for survival. And these are the only two columns that I need to do my survival analysis. Now you'll see that this overall survival column, which I mentioned is numbers and days, has this peachy orange header. You'll see that this is just a nice, quick and easy way to distinguish different columns. Any numerical column, so we also have age at diagnosis, which is, of course, all numbers for age, and total non-silent mutation, which is also just numbers. All these numerical columns have this peachy orange header, and that's just a quick way to see what different kinds of headers we have. Okay, so we have all of our samples in here. We have these different factors or annotations that we've added in, and we have survival data. Now, before we even get started looking at our copy number and mutation data, we can actually start to do some survival analysis right in this front page. So one thing I like to do when I first begin a study with survival is I like to first validate that everything is behaving the way I anticipate it to by looking at tumor stage. We would anticipate that those that are diagnosed at stage 4 will have a significantly worse overall survival than those diagnosed with stage 1. And so that's just a nice quick test to make sure everything's behaving normally. So what I can do is go over here to tumor stage, right-click, and go down to this KM plot, which is a Kaplan-Meier plot. Okay, right now it's empty, but you can see there's a drop-down box here that says select time factor. And when I click this, it's only going to give me options for those column headers that were that peach-orange number column. And I want to click the one that's for my survival, which is overall survival is what I've called it. And as soon as I clicked on it, it loaded up our Kaplan-Meier plot. I'm just going to enlarge this slightly so we can see it a little more. So we have all these different bars. If we hover over the bars, we can see which each one is. So we have the worst is stage 4. This is stage 2, stage 3, and stage 1. And so we can see that it looks like stage 4 did perform the worst, and stage 1 the best. It looks like 2 and 3 are a little flip-flopped here, but that could have a lot to do with the number of samples within my data set or just how this data is behaving. Of course, I would really like to know the significance of all of this, so I can actually just click over here on this button that says significance to evaluate the p-values. And we can see that the stage 4 is statistically significantly worse in terms of survival compared to all 3 stages. Now, if I wanted to export this out for a publication or a presentation, I can simply copy and paste this table, and I can save this figure. So it's really easy for me to get, high-quality figures in just a second. Okay, so this all looks good. So now I want to know if there's anything else that I wanted to evaluate. And one thing I was particularly interested in with this data set is this colon polyp diagnosis. Now you can see I don't have data for every single sample, but I do for some, and I know that this is a risk factor for COAD. So I was really curious if it had any impact on the overall survival. So if I, again, right-click and go down to the KM plot, you can see now the overall survival button has already been selected, so that's preloaded in. I didn't have to reselect it. And now if we go over here, we can hover over it and see that the presence, so the presence of colon polyps has overall worse survival, whereas the absence of colon polyps it has a slightly better survival. If I look at the significance, I see that this does not, however, reach statistical significance. My p-value is 0.18. So interesting, but not necessarily significant. And I could look at much more of these if I wanted to. I could look at, the anatomical site or histological site, but that's for a better discussion. Okay, so now that we've evaluated that, what about the copy number or mutation event? So I'd like to look at that data, so let's go ahead and we'll click View. And while that's loading, I want to discuss with you about different ways for us to process, evaluate our data. Okay, so when we're doing survival analysis, there's 2 different approaches. The first is an unsupervised or indirect approach, or what I would call a fishing expedition. And in this approach, what we're going to do is we're going to identify changes of interest just through our routine analysis, and then we can later test to see if there's any survival differences. The ease of this depends on the number of samples in the study, as well as the number of samples that are in each survival group, alive versus dead, and just the overall variability within our copy number profiles. And so like I said, sometimes it can be easy, sometimes it can be really hard. It just depends on your data set. Okay, so to refresh your memory here, this is our results tab. This top panel is showing us our summary or aggregate view. You can see blue for gain, red for loss. If we go look at the bottom panel, we have each of our individual samples with their names located at the left. You can see, again, blue gains and red losses. We also have mutation data loaded in. So you can see here are all of these little symbols on the top indicating different types of mutation variants. And so, like I said, right now we're just looking to see if there's anything that catches our eye, if it's something that we would like to see if it has an impact on survival. And one thing that I noticed was it seems like there's a very frequent gain on chromosome 13. And if you look at some of these individual samples, it does appear that it spans nearly the whole chromosome in those that are gained. So my question was, does gain of the entirety of chromosome 13 have an impact on survival? And so in order to find out, we have to do a multi-step approach. So we're going to go back to our data set. First, we need to identify which samples actually have a gain in chromosome 13. And so I'm going to use this query button. And I'm going to click the button here, and a pop-up window will appear. There we go. And now we can specify if we want to do it for genes or regions. I'm going to do region here. This is for chromosome 13. Now, I'm going to cheat just a little bit. So the probes on chromosome 13 don't start right at the beginning. They start in just a little bit, so I'm not going to start looking right at the very beginning, but in just a touch. All right, let's make sure I typed my numbers in correctly. They look good. Okay, and then I'm going to hit completely covered because I want it to be gain of the entire chromosome, not just a region. And then I'm going to hit run. And you can see really quickly Nexus looked at all of the samples here and asked, do you have a change across this entire chromosome, and if so, what is it? And we have our results here. So I'm just going to sort. Okay, and so now we can see that there are a number of samples that have copy number gain, some with and some without allelic imbalance. So I'm just going to select those samples. So I'm going to click on the first one, and then go down to this last one here, hold down my Shift key so I can highlight this subset, and then hit select. Okay, so I've now selected all of the samples that have a full chromosome gain. But I need to apply this to my back page here, the data set tab, so I can actually run my Kaplan-Meier analysis. So what I'm going to do is I'm going to click Apply Selection. And when I do that, the back screen, only some of these are going to be selected. So let's hit Apply Selection, close this window. What you can see here now is that only some of those samples have been selected, and those are all of the ones that have that full gain. Okay, now I'm going to manually add a factor. So I'm going to go to Factors, Add Factor, and I'm going to call this chromosome 13 gain. And whenever we add a new factor, it's going to add to the far right. So all I'm going to do is just click and drag this over to where I'm working. Okay, I'm going to sort over here based on those ones that are selected so that I can now add these samples in. And a little shortcut, there's a button over here that looks like a little table. If we click on that, I'm going to click Allow Column Selection. And what that's going to do is it's going to allow me to select individual cells. Okay, so what I'm going to do now is I'm going to label these first set as a Y because they have been gained. So if I select and then right-click Set Value, Y, so those all do have a gain. Now all the rest of these do not. Okay, and so I'm going to label all those N. Okay, and so now everything has been filled in with a value. Now I'm going to do Control-A, select, to select all of the samples here and run my Kaplan-Meier analysis. So again, right-click here, Kaplan-Meier. So now we can see if you have a gain of the entire chromosome 13. So yes, you do have the gain. You have an overall worse survival than if you do not. If we go to significance, we can see that this is not quite significant, but really close. So it could be something of interest, but it didn't quite reach statistical significance here for us. So again, we're really just doing a fishing expedition, but what if we want to try something a little bit more direct of an approach? So if you have something listed as a factor on this page, you can also run comparison testing. And you'll notice that one of our factors, the event factor, is alive and dead. So what if we compare, group together and compare the alive samples versus the dead samples to see if there is any sort of difference in their overall copy number profiles that might help lead us towards the direction of something to evaluate further. So to do that, we're going to click Comparison, Add, and now we're going to select our header, which was Event. And you can see here we have alive versus dead. Okay. And I'm going to do a paired analysis, and I'm going to make alive my baseline and click Finish. Okay, so we have our category is event, dead versus alive, and we're going to run a Fisher's exact test to compare these two groups. So I'm just going to highlight it and click View. And so what we're doing now here is we're looking to see if there's anything that's statistically significantly different between these two groups. All right, so I'm going to go to Genome here, and we'll look at the visual display of our results first. So I'm just hitting this slider to zoom in. And I'm also going to change my view here to peaks only. This gives me just the highest peak of change. Okay, so what you can see here is we have 29 samples that were in the dead group, 191 in the alive group. And you can see that overall they look pretty similar to one another. If we go to this significant line, we can actually see the areas that are statistically significant between the two groups. So it looks like there is a lot of loss of 1p. Within more loss of 1p within the dead group versus alive. There's this peak of gain right here on chromosome 5, peak of loss right here on chromosome 6, a little bit of variability in 7. It looks like maybe this loss extends a bit further in 8, loss of 16, loss in 18, and then this tiny little gain on the X chromosome. So not a lot of changes, but a little bit of something. So let's now look at our Regions tab, which is our table view, to see if there's anything of interest in here. And so you can see here now we have, on this table, it's showing the actual region that was identified as being significantly different, site of band location, type of change, length of change. We have the frequency in the dead group versus the frequency of the alive group, and then that difference between the two groups. We have our statistics, both our p-value and our q-bound, percent TMB overlap, and this one's important because it's going to tell us if this is a common variant or something that might be a bit more rare. And then our annotation for G. All right, so you can see here I have a lot of different kinds of change. I'm really more focused on the copy number changes for this particular evaluation, so I'm going to sort by that, and then hit down the control and sort by region so I can have these in some sort of order. So let's scroll down, and what I noticed is that, especially for the gain one, you can see these are all 100% DMV overlap, which means that they're really common changes, so not likely to be something pathogenic. All right, and if we can just scroll down some more, I did see here this one here that's 0. So this is one that is not known to be a common copy number polymorphism. And I would like to see now if this particular loss on chromosome 8 has any impact on survival. And so you remember we have to now add it as a factor. So we have a little shortcut in this particular view. So if we right-click on this, we can actually go right down here, query all process samples in project. Okay, so this is going to shortcut us right to that query window that we use to identify our samples of interest. And so again, we're going to sort here. We're interested in the ones that have loss. So again, I'm going to use my shift keys to highlight and select all of those that have copy number loss. There's a few more up here. Let's add those ones in. And again, I need to hit Apply Selection to apply it to my main page. I'm going to click Apply Selection, and then I'm going to X out of these windows. And so you can see here not only that subset has been selected. Now again, I need to add a new factor. We're going to call this one loss chromosome 8. So this is just that for that region. And again, always adds to the right. Click and drag over to where I'm working. Sort. Okay, and now we can add in our annotation here. So we want to make all the ones that are selected are yes, they do have that loss. All the ones that are empty do not. Okay. And then I'm going to select all so that we can run our Kaplan-Meier analysis. It's really important to make sure you have all of the samples selected that you want to be included in the analysis, because you can select only a subset of samples if you would like to for your Kaplan-Meier analyses. For this particular instance, I really want to evaluate them all as sets. Okay, so now I can right-click on our factor here, go to the Kaplan-Meier plot, and so now we can see here that loss of chromosome 8 has poorer survival as compared to those that does not have the loss of that region. And if I click on significance, we can see that it just barely reaches statistical significance. So our p-value is 0.049. So this was a way for us to identify something that does have a statistically significant difference in terms of survival between the two groups. However, this was still, we're still doing a little bit of a fishing expedition. It's a bit more direct. But there's also the fact that we're really only comparing alive versus dead. We are not taking into account with this analysis the actual number of days for our overall survival. And so there is another tool that we can use that will give us a more direct approach, and that's survival predictive power. So in order to run that, we're going to click on our overall survival column. You can see when I right-click here, I actually have a little bit more in my drop-down box, and I'm going to go to survival predictive power. Okay, and while that's running, let me just explain to you what we're doing. Okay, so now we're going to go for a more supervised or direct approach. And what we're going to do here is we're going to test each position in a genome for events that could be predictive of survival. And this is based on both the alive versus dead, but also the length of survival for each sample. And so what we're going to be able to do with this is find significant regions with ease and easily exclude some of the noise that we might be seeing. So how does this actually work? Okay, so I'm just going to give you an example to help explain. So imagine, if you will, that we're looking at a region of chromosome 1. Okay, and we're looking at a snapshot of 7 different samples. So here we've got these 7 samples. They all have a gain within some part of this larger region of chromosome 1. We can see here that some of these have an overlapping region of gain, so that's this part in between these 2 hash bars. And some of the other samples do not have overlapping gains. So 7 and 3 have no gain, and 7 and 3 have no gain within the region. 5 has no gain whatsoever within this entire larger window. And for each sample, we also have survival associated with it. So what we're doing here now is we're going to group together those that are sharing the event, and then calculate their average survival and do permutation testing to see if there's a significant difference. So in this group, we have 4 samples that have this overlapping change. Their median survival, here mean survival is going to be 10, 5, 15, and 20, or an average of 12.5, versus those that do not have this event have survival times of 90, 60, and 80, or an overall average of 77. And so with permutation testing, we can then determine if this is statistically significant from one another, and if so, we'll be able to view that in our table. So what we're doing now is we're doing this calculation for the entire genome. Okay, let's just see. Oh, here we go. It just popped up. Okay, so here are our results. We have a regions tab, which is going to explain all of our areas of interest. We also have a genome and chromosome view for both copy number and allelic events. We can also visually observe what's going on. And so what we're looking at here now is we have all of the regions of change that were identified as being significant. We have the type of change, length of change, number of genes. We have this column called in. This is the number of samples that have that change, so that are in the group, and their median survival time. We have out, which is the number of samples that do not have that change, and their median survival time. And then we have a difference here, which is showing the differences in survival. Now we have our statistics after this. We have our p-value, our permutation p-value, and then our q-bound, which is testing for multiple correction. And finally, we have our gene annotation. So you can see here we actually have a lot of results. So I'm just going to narrow this down a little bit. I'm going to go for peaks only, so I can get our peak region of change. And I'm going to change my p-value threshold to be a little bit more stringent. And so you can see here we have many different regions potentially of interest. So let's look at our overview of this visually so we can see what we have to look at. Okay, so when we look at our whole genome view tab, the top panel here is showing us just our genome annotation. And then we have two panels here. This uppermost panel is going to show any differences based on gain. And this bottom panel is going to show differences based on loss. Our y-axis over here is going to indicate the length of our study. So this study lasted about 4,000 days. So you can see that the uppermost value that we can have is 4,000. And now what we can do here is we can see what it looks like when we compare the in versus out groups, how their survivals are different. So for example, if we look at 1Q, we can see that the out group, which is black here, they do not have the change, they're diploid, has an overall survival of 3,000 days. However, the loss group, which is way down here in red, has an overall survival of 185 days. We can also zoom in to view this a little bit more closely, so you can see this here. We can see the black bar here for the samples that did not have the loss, and then the red bars for the samples that do have the loss. So this is just a way for us to visualize what we're observing. So now let's go in here and see if we find anything that might be of interest. So I first like to always look at these Sanger cancer gene census so we can see if there's any known cancer genes. And one that caught my eye is this beta-catenin, which I know is involved in CLAD. There's also one of these here which has a very high significance value even after multiple testing corrections, so this one could be really interesting. Now, one of the extra great features about this particular tool is that we can really easily add factors to find our Kaplan-Meier analysis. So we already know that there's going to be a difference in survival for this, but I want to know how great that survival difference is. So we can look at this numerically here and just see that there's 15 samples that have this loss. They have a median survival of 227 days. 205 samples do not have this loss, and they have a median survival of 3,041 days. So let's click Add a Factor here, and let's view our Kaplan-Meier plot for that. So when I click that button, I now added a new factor to the far right. We have copy number loss at our region for beta-catenin. And if I right-click right here, I can now very easily do a Kaplan-Meier plot just for the specific event. And so you can see here it looks really dramatic. So hover over this, we can see that this is the samples with the loss versus those without the loss. And our significance value is 6 to the negative 8, so really significant. So this is a really direct approach for identifying regions of interest that have differential survival analysis. All right, so to wrap up, today I showed you how to add annotation to your data set, and also how the addition of survival data can really enrich your overall evaluation. We can use indirect methods to identify significant regions for survival analysis, and this is by identifying a region of interest and then evaluating its survival in the data set tab. We can also use more direct methods, like the survival predictive power tool, to identify statistically significant regions which affect survival. And then we can use that one-click Add as Factor button to generate a factor and get our Kaplan-Meier curve. Okay, and so with that, let's wrap up, and I'm going to hand it back over for our poll question.